Hi, my name is Viviana Hermosilla. I am a bioengineer. I have a master's degree in biochemistry and bioinformatics, and I am a PhD student. First of all, I want you to meet my campus. This is the view of the University of Concepción from my window. As you can see, it is almost empty. There is no one here. This is because the university remains closed by this date. Undergrad students went on strike a few weeks ago because they want a tuition freeze. The university has kept the tuition fees frozen for 3 years, but now they must be increased in order to support its expenditures. Because this is a private university, although it receives some government contributions. Students are very angry, so they occupied some university buildings. Authorities decided to close the university in order to allow cops to get in and take the students out of the buildings, and just then the classes can be restored. We cannot attend our labs either because health insurance provided by the university is not available while the campus remains closed. So if we get hurt because of the manifestations, they wouldn't take responsibilities. This is the building where I work. It has 4 floors. The first and second floor belong to the Department of Biochemistry and Molecular Biology, and the third and fourth floor belong to the Department of Cellular Biology, where my lab is. And now we are going to gather outside to see if we can get in. Last but not least, please allow me to introduce my lab. This is the Axonal Guidance Lab, directed by the PhD Teresa Caprile and Concepción, PhD Hernán Montesino. As you can see, it is quite small, but we have almost everything we need, such as refrigerators, a safety cabinet where we manage nucleic acids to avoid contamination, an incubator where we brood our eggs because chick embryos are our model of study, a cell culture room with a laminar flow cabinet, a light microscope, and a CO2 incubator, and a room that it is used to electroporate animals and dissect animals. Unfortunately, I am totally alone by now because my professors and my lab mates are attending the annual meeting for cell biology in Puerto Varas, but I had an idea in order to remember them. So here is Amerita Vera. She's a master's student. Melissa Gonzalez, she's also a master's student, and they are both classmates. And over there we can see Karen Stanik. She's a PhD student as I am. We also count with a technician who keeps everything clean and ready to use. And here is my boss, PhD Teresa Caprile. This is the view of the Axonal Guidance Lab. In the central and peripheral nervous system, a heterogeneous group of proteins constituting the thrombospondin superfamily provides a cue for axonal pathfinding. Among these proteins, we can find SCO-spondin, a novel relative of the thrombospondin family involved in neuronal development by modulating cell aggregative mechanisms. SCO-spondin is a glycoprotein secreted by the subcommissural organ, an ependymal differentiation located in the diencephalic roof, beneath the posterior commissure at the point where the axons cross the midline. This protein has several conserved domains in a unique arrangement in mosaic, such as 26 thrombospondin type 1 repeats and 9 low-density lipoprotein receptor type A domains. My job consists of determining the function of the thrombospondin repeats and LDL receptor domains on neurons that cross the midline through cloning, expression, and purification of peptides whose sequences include highly conserved amino acids, and subsequently dissect the signaling pathway involved in mediating this effect.